This video is looking through the B6 distance learning questions so that we can green pen and looking at the mark scheme. So this picture first of all shows us an ancestor of elephants, so I'm not going to try and start saying the name for, and the modern African elephant. The ancestor lived 35 million years ago. It's got this long nose, and the African elephant has the trunk. Both of the species reach up to trees to get leaves. In the 1800s, Darwin and Lamarck had different theories about how the long nose of the ancestor evolved into the trunk of the African elephant. And the first question says to use Darwin's theory of natural selection to explain how the elephant's trunk evolved. We've got 4 marks for this, and we've got 1 mark for saying that there's variation in the population, which could be caused by sexual reproduction or mutation. 1 mark. The longer nosed individuals get more food, therefore they live longer and reproduce and pass on those genes, those alleles or that DNA to the offspring. Okay, so that's our 4 marks, 1 mark for each line of writing on the page. Okay, let's move on. Lamarck's theory is different from Darwin's theory. Use Lamarck's theory to explain how the elephant's trunk evolved. So the ancestor stretched its nose during its lifetime to reach the food. We know that that's not the case. Its nose is not going to get longer because of overstretching, but that's what Darwin believed at the time. And then it's believed by Darwin that that passed on stretched nose went to the offspring. So where it had stretched and had that environmental change over its lifetime, that new feature went on to the offspring for the 2nd mark. In the 1800s, many scientists could not decide whether Lamarck's theory or Darwin's theory was the right one. Give 2 reasons why. There was insufficient evidence. At this time, people didn't know about genetics. They didn't have a full understanding. They were just looking at physical characteristics of individuals. And that leads on to the 2nd mark. The mechanism of inheritance was not known, which basically just means we hadn't discovered DNA. We didn't know the building blocks of life and how everything came together. Before the 1800s, many people had different ideas to explain where all the living things on Earth came from. What idea was this? And the idea straight from the mark scheme was that God made all living things, or you could have written in there creationism. Okay, so that's the end of question 1. We've got through that quite quickly actually. Question 2 is a bit of a nightmare. Okay, so don't worry if you find it too difficult. We will get through it together as best we can, trying to understand and unpick this question. So question 2, the diagram shows how scientists can use genetic engineering to produce human growth hormone. Okay, a lot of hormones are produced in this way. So we've got here, we've identified the gene for the growth hormone, and you can see enzyme 1 snips it here to get that gene out, to remove the gene from human DNA. Over here we've got a plasmid from a bacteria, and we can see there is a gene here for the ampicillin resistance, okay, which is an antibiotic, so it's being resistant to that antibiotic, and the gene here for tetracycline resistance, okay, over here. Now enzyme 1 is also going to cut through that gene, and we can see it's opened up that gene, and enzyme 2 is putting that gene for the growth hormone that was taken from this human DNA into the plasmid there, but it's chopped in half through that tetracycline resistance gene, which is also resistance to another antibiotic. The bacteria which is not resistant to either of the antibiotics, so the bacteria was not resistant to either, and then we've got the bacterium with the plasmid with the gene for the growth hormone, okay, and it's releasing growth hormone, which we're then going to be using, okay, and then we get cell division and a clone of all that bacteria. Okay, so we've used an empty bacteria here which wasn't resistant, but the plasmid that is going into it has the resistance for this one and has the resistance for this one, but it's being chopped in half. and the growth hormone's been put in that place, okay? So, human growth hormone is made by the pituitary gland. The human DNA containing the gene for growth hormone can be taken from a white blood cell. Give a reason why the gene does not have to be taken from the cells in the pituitary gland. And that is because the white blood cell, they have the same DNA, genes, chromosomes, whichever word you choose to use, that you would find in the pituitary gland. Okay, so most of our cells, obviously not red blood cells because they don't contain a nucleus, but they all have the same genetic code. So that growth hormone gene can be found anywhere in any of those cells that have got the chromosomes in, got the DNA in. Okay. The figure above shows that the plasmid contains two genes for antibiotic resistance. A gene for the resistance to the antibiotic ampicillin. Sorry, I don't know how to say these words properly, but we'll go with it. And a gene for the resistance to the antibiotic tetracycline. Okay, so we've got the resistance to two particular antibiotics. We may have to come back to this page. I'll just put it to the side here. Okay, next bit kind of goes down the line of what we know from B2 in digestion. Explain how the structure of enzyme 1 allows it to cut the gene for tetracycline resistance, but not the gene for the ampicillin resistance. And that's because of what we know about lock and key theory. The enzyme's active site, notice it's underlined, that means we have to use the words active site, has a specific shape, 1 mark. The two antibiotic resistant genes have different bases, so they're actually different bits of protein, so they're made up of different bases. Again, bases is underlined, another mark. That's probably the most difficult mark to get in this question. And only the tetracycline resistant gene fits or is complementary to the active site of the enzyme. So that's why the enzyme is able to chop that part, because it's the right lock and key for it. It's complementary. Okay, hopefully that makes sense. Now I thought this bit got quite tricky, okay, because we've really got to think about this. So I'll do my best to explain. In the final step of the diagram above, that means the diagram on the previous page, very few bacteria take up the plasmid containing the gene for the growth hormone. Some bacteria take up an unmodified plasmid, so the original plasmid. Most do not take up a plasmid. Complete the table below for basically those different options. Put a tick in the box if the bacterium can multiply in the presence of the antibiotic. Put a cross in the box if the bacteria cannot multiply in the presence of the given antibiotic. Here are the results. So, let's go slowly with this to make sure we understand. The bacterium with the plasmid with the growth hormone, okay, bacteria can multiply if there is the ampicillin antibiotic, and that's because there is ampicillin resistance, as we saw in this picture over here. Okay, there's ampicillin resistance. That means the bacteria are resistant to that antibiotic, therefore can still multiply. But the tetracycline resistance, this gene has been damaged where it was cut open to put in the growth hormone gene. So in the presence of that, they can't multiply because they haven't got the resistance to it anymore, because that gene has been damaged. Hopefully that one's explained okay. The bacterium without a plasmid, the bacterium can't multiply because neither of those resistant genes are present. Easy enough. And the bacterium with the unmodified plasmid means that that section of gene there has not been damaged because it's not been modified. So imagine that, like, it looks like this, where it hasn't had the enzyme put in. So both of those resistant genes are there. Therefore, if we've got an unmodified plasmid, the bacteria can multiply because they're resistant to the antibiotic. Yes, I think I've explained that correctly. In fact, I know I've explained that correctly because I had a little practice earlier. Okay, question D. By the way, that was a super hard question that's probably going to be found on a triple higher paper only. but that was the point of these videos. These distance learning questions were intended to be a real challenge, designed to push those people who really want to improve their exam technique and really shine in those exams. So that's why I've chosen such difficult questions. So don't let it put you down, just kind of go, okay, well, I kind of get my head around it now, or if I saw something similar, I think I probably could give it a better go than prior to doing these questions. Anyway, last question. The figure above shows the bacterium containing the gene for human growth hormone multiplies by cell division. This produces a clone of bacteria. Explain why all the bacteria in this clone are able to produce growth hormone. Okay, so the clone is produced by asexual reproduction. There's no copying of information from two different sources. It's all come from that one individual, so that's why it's got exactly the same genetic information. Therefore, it goes on to the second mark, all the DNA or genes, you could say that one, are copied, and then as a result, every cell receives a copy, or every gene, or the plasmid. Okay, so it's just taking that information like a photocopier, giving all of the genetic information to the new cloned individual bacteria. Okay, right, hope that all made sense. If it didn't, send me a message on Show My Homework and I'll try and make as best as I can explanation. Okay, right, enjoy your day, thank you, bye.